So today on to Rare, we welcome Adrien Lemoine. So welcome, Adrien.
Hi, Laura.
Adrien is the co-founder and CEO of Bloomsbury Genetic Therapies, which is a spin-out biotech of University College London and focusing on the development of treatments for patients suffering from rare neurological and metabolic diseases, with a pipeline of first or best-in-class gene therapy programs. So welcome, Adrien. Thanks for your time today. Would you be able to tell us a little bit about yourself and your journey so far?
Sure, pleasure is all mine, Laura, and thanks for having me today. So yeah, I've got I think, slightly original journey. I was trained as an engineer in France, where I'm from, as I'm sure your audience will have understood by now. With this accent, it will never leave me, I'm afraid. And moved very early on in consulting in the field of biotech. So I was supporting a lot of biotech, small companies, entrepreneur. Gave me a taste really for biotech, and I've never turned my back to it. And I've been working for the past about 20 years of my career now in biotech and pharma. I moved to big pharma following my consultancy experience, went to GSK for about 10 years. The last 5 were in the group we set up back in 2008 in rare diseases. So there was GSK dabbling for the first time in a thought through fashion in rare diseases, with the idea to leverage all our GSK capabilities and all of their disparate program in rare disease and put them into a single unit with a focus to really bring them to patients by leveraging all of the GSK clouds. It was a great experience, which gave me really a first taste for rare diseases and gene therapy, because it was back then one of the first kind of big pharma to really invest heavily in gene therapy. And that never really left me. I did a short stint at AstraZeneca following my time at GSK, but then when a company creation project, which came to be Orchard Therapeutics, got on my radar with a few ex-GSK colleagues at the helm, I really went for that opportunity and was lucky enough to be one of the first members of Orchard, which has been a great scientific success on many programs. That a few of them are commercial now, and gave me a fantastic opportunity to be involved even more deeply in the field of gene therapy and rare diseases. And I left Orchard a couple of years ago to found Bloomsbury Genetic Therapies, which also have been a fantastic adventure. I mean, the project was coined about 3 years ago in collaboration with our current seed investor, the UCL Tech Fund, which is managed by Adrien VC, a UK-based investor, which is dedicated in funding innovation out of UCL. And we thought there was a better way or a faster way to do gene therapy development in rare diseases. That's what I was seeing that firsthand at Orchard, being on the front line of all the challenges that were thrown at us from regulators, the challenge of making gene therapies at the right quality, in the right volume, and the time it has taken really to get the first gene therapies approved. And we're talking about 30 years between the idea of gene therapy as a therapeutic option to the first approvals. And in rare diseases where there's nothing else, but where the promise of gene therapy is so important, because you go after the root cause of the indication, you fix the 40 gene, it should be faster, it shouldn't take that long, and patients really can't wait that long for the majority of the rare diseases out there, which are sadly often lethal in the first couple of decades of life. So I was thinking there must be a faster way to go about gene therapy for rare diseases, now that we have a few approved products, a few pioneers who made it through approvals, and Luxturna, Zolgensma, we have Roctavian now, Elevidys, and a few CAR-Ts and a few HSC gene therapy products. Now that there's a few approvals and that regulators are comfortable with, obviously, with approved programs, I thought there must be a way to leverage the learnings from the first approvals, leverage some of the building blocks of those therapies to avoid having to reinvent the wheel and spend another 10 years in development, and be much faster to translation to the clinic, and then much faster to approval. Obviously, the end game being having an approved drugs. But how the idea of Bloomsbury kind of germinated, as I was discussing with investors at Adrien VC, and how I came to meet my co-founders at UCL, who are all academics, and we're coming at it at Bloomsbury with a similar angle of obviously a need to bring something quickly to patients by leveraging what has worked, again, for that idea of speed and efficiency. So that's how we came to create Bloomsbury, and I was very excited to have a shot really at starting from the scratch on that vision of being very pragmatic and very, very efficient in terms of time, money, developing gene therapy for rare diseases. So building really on all the knowledge I've acquired at Orchard in particular, but also at GSK and all the contact I have in the industry to try to do that 2.0 gene therapy company that can move faster than the pioneers, where there are a lot of hurdles to overcome for the first time for the benefit, hopefully, of company like us now.
And how did you feel about making that plunge from being a chief business officer to a co-founder and starting something completely from scratch?
Yeah, so it's a great question, because I had a whiff of that a little bit at Orchard, because I think I was one of the first few employees, so starting very early, but it's quite different when you start from the get-go, because you can really set the vision yourself. And I had a very clear vision for Bloomsbury to be pragmatic and efficient and move fast. And I think we've been quite successful and vindicated in our vision in the 1 year, more or less, of operation we've had since we closed our initial funding round last year. But of course, it comes with a lot of new challenges for a first-time CEO, which everyone in my position has faced, from having to manage a team across a variety of functions where you're not yourself the expert, to fundraising, which obviously is a bread and butter, and in particular these days where we all struggle to raise money in biotech. It brought a lot of new challenges. Some of them I was anticipating, some of them were new, but I've been lucky to be held by a very experienced board of directors with a great chairman, Frank Armstrong, a lot of experience, and a couple of independent directors. So they've been very helpful in helping me navigate the first few quarters as a first-time CEO.
Yeah, no, that's great. And so let's talk a bit more about Bloomsbury Genetic Therapies. So what is Bloomsbury all about? Like, what's the purpose?
Yeah, so it's really plugging that gap of delivering on the promise of gene therapy in rare disease, which conceptually should be fast and bring essential drug to patient quickly, but has been difficult for now. So it's really about trying to solve that equation by leveraging what has worked already in the field, and in the particular flavor of gene therapy we're going after, which is AAV gene therapy. It's in particular using well-known capsids, because if you look at what has been challenging for AAV developers over the years, is two things. It's safety, first and foremost, and sadly we still see trials where the patients suffer from side effects. Some of them can be quite severe because of the high dose of AAV being used sometimes. And the other one is CMC. So manufacturing gene therapies has been a challenge for everyone, and it's kind of a well-known trope now in our field. There's also that trope which says that the process is the product, which is less true these days, but still bear a lot of weight and validity. If you change the way you make your AAV, you might change the product itself. So of course, the way you make it the first time, when you do a first-in-man trial, is not always the same way that you make it when you prepare for commercial, because the process has changed and evolved. So that has set back a lot of developers, going back to more or less square one, and need to redo trials with an improved process. So if you look at safety and manufacturing, and you look at what is the common denominator of both aspects in AAV gene therapy, it's the capsid. It's the capsid which will dictate most of the safety issues you have in your trials, and it's the capsid which is the main challenge, or the main which will define the way you make really your AAV vector. So that's why at Bloomsbury, what we're trying to use is capsids which are already been de-risked, i.e. they are part of an approved product, like we're using AAV9, which is part of Zolgensma, for instance, using AAV2, which is part of Upstaza, both drugs approved worldwide for Zolgensma, in Europe for Upstaza, and we're using LKO3, which is also a capsid that is part of an advanced hemophilia A program led by Roche and Spark. And in each case, those programs have shown that the capsid are relatively safe. The dose at which you can use them, which provide safety, you know what they distribute, you know you can make them at the right titer, the right volume. So all of those all-important elements on which a lot of first-generation companies have stumbled on are de-risked somewhat if you use those well-known capsids. So that's the key building block of our approach, well-known capsids, but obviously peppered with innovation on the choice of transgene, the choice of promoter. So we have a variety of patents protecting different aspects of our vectors. So the idea really is to move much faster with those known capsids, and that's what we've been delivering so far, touching William on the first year of operation, and that's what I intend to deliver for Bloomsbury in the coming next years of operation, bringing drugs to patients fast, and delivering on the promise of gene therapy for rare diseases.
No, it's very exciting to see. And you did announce some very exciting news recently. So can you tell us a little bit more about that?
Yeah, we've announced very exciting news on the 6th of September, where we've announced that we are developing or expanding one of our programs called BGT-DTDS, which was developed and is developed primarily for a very rare childhood disorder called dopamine transporter deficiency syndrome. DTDS is the acronym for the disease. It's a very severe childhood disease where kids are born without the ability to express the dopamine transporter protein in the brain, in the neurons, which cause a variety of complications, primarily motor function issues, and then wider CNS disorder, and kids die usually at the beginning of the second decade of life, and there's no treatment. It's relentless disease, and there's a progressive loss of motor function that is very harrowing, and the hallmark of most rare disease is there very strongly for the DTDS. So that program is using an AAV2 vector, and we're injecting directly in the brain of patients an AAV2 that carries a copy of the functioning dopamine transporter gene to restore the expression of that protein in the brain of patients. And we thought, well, actually, there's another disease where patients lose the ability to make that transporter. It's Parkinson's disease, where in Parkinson's disease, damage caused by alpha-synuclein accumulation progressively destroys your dopaminergic neurons, and you lose a few things. You lose the ability to make dopamine in the first place, which is why patients take oral L-Dopa to top up dopamine levels. But you also lose the transporter. So patients end up in a state called levodopa-induced dyskinesia, where they either have too much dopamine or too little, and it's becoming much harder to manage, in particular the motor symptoms of the disease. So our approach is to use the same vector as the one we developed for DTDS, with the view to rebalance expression of the transporter as the degeneration progress in Parkinson's disease, and reestablish dopamine homeostasis in the brain of patients. So it's a very interesting case of starting really a program in a ultra-orphan, simple biology disease like DTDS, and use it as a way to de-risk a much more complex disease like Parkinson's disease, etiology and is much more complex, but which involve that, so dopamine transporter, as a key tenet of its evolution. And we just completed the in-life experiment in a rodent model of Parkinson's disease using that vector. We're very excited to report that we have a profound effect on levodopa-induced dyskinesia, and we've seen as well some effect on anxiety behaviors in the rats. So it's very exciting because we may have a very novel approach to go about Parkinson's disease, motor function and non-motor function symptoms. We're going to elucidate the pathway, the way the treatment works a bit further in upcoming studies. And we believe we might have a potential disease-modifying treatment in our hands with this program. So we just revealed it. It's synergistic with the TDS, which is great because it's same vector, same CMC development. The biology of the TDS has some parallel with the one of Parkinson's disease. So one disease help each other. So it's a very nice case of going from rare to non-rare, which we are keen to explore ourselves and potentially as well in partnership with companies who have a lot of experience in Parkinson's disease as well.
Well, congratulations. It is very exciting, and for all those patients out there as well. So what can we expect then from Bloomsbury in the next sort of 12 months?
Yeah, there's a lot. I mean, we've done a huge amount in our first 12 months of operation with a limited amount of capital and a small team. There's 6 of us in the team. That's about it. And leveraging, obviously, our colleagues and collaborator from UCL who provide fantastic research activities, both clinical and non-clinical, to the company. But expect more of the same in the next 12 months, more very efficient and productive operations. First and foremost, fundraising. So I'm actively fundraising now for the company, following the first 5 million we have in the bank, and we've done a lot with those 5, and I want to do even more with the next round of funding that I'm currently working on. But with money in the bank, we expect to meet a few very meaty milestones in the next 12 months. So for our lead program for the liver disorder, OTC deficiency, we expect to get our clinical POC in hand. So it's a very exciting program because we're starting the trial in a matter of weeks now for the first-in-man phase 1/2 trial in the UK. And it's a disease where you have very quick efficacy readout. So within the first few months post-treatment, you get to see the efficacy of the treatment and, of course, the safety. So we'll have those first readouts in hand in the next 12 months and get to really full clinical stage status for the company. For our second program, so for our DTDS program, after the great success we had with the MHRA earlier this year, where we really, again, walk the talk of Bloomsbury positioning by going to the MHRA with efficacy data in mice in hand, making the case that AAV2 for brain administration has been done many times, and that there's really nothing much we would learn, or the field would learn, by doing further exploration of distribution, for instance, of the brain, of the vector in the brain. We made the case as well on safety, and we got the support of MHRA to go straight from knockout mice efficacy data to clinical trial, which, to my knowledge, is probably one of the first time a company has been able to move straight from mice to human without stopping on NHPs or dogs for safety and distribution. And again, a testament to our approach of leveraging precedents and success from the past, from Upstaza, other intra-brain AAV2 treatments, which have already de-risked this approach for us. So for that program, expect in the next 12 months that we're going to gear up for that first trial, which, by the way, we intend to lead a single trial, a single phase 1, 2, 3 trial in a very compact design. Again, following my vision for Bloomsbury to be very efficient in bringing drugs to patients quickly by really front-loading the thinking, so that we can really be much faster when in the clinic, and having a single trial is obviously much quicker than having 2 consecutive trials, which has been kind of the more usual way to develop gene therapy. But now we have the knowledge, the experience, the regulators as well have the appetite to welcome developers like us to come up with more compact designs. So expect us to gear up for that first in man, and in particular the CMC campaign we need to start once we are in funds to procure GMP vector or that trial to start. So the sister program, Parkinson's disease, so expect in the next 12 months more preclinical data. We want to bring the vector in different rodent model of Parkinson's disease to elucidate in more detail the way it works, and as we believe as well elucidates a true disease-modifying effect on Parkinson's disease. So there will be more of that in the next 12 months. For our last 2 programs for Niemann-Pick disease type C, NPC, and infantile neuroaxonal dystrophy, INAD. Here again, we just scored great support from the MHRA, where we managed to convince the MHRA that we only need to do very shortened safety and distribution studies in rodents. Again, a game-changing approach compared to doing them in NHPs, which is much more expensive and long studies to run these days as well, where NHP are hard to source for a variety of reasons. So with that success in our back pocket, we're going to meet and engage with the FDA in October on those programs, for which we've secured a Type B and pre-IND meeting, confirmed the plans for the remaining IND enabling work, which we hope to be similar to the one obviously the MHRA has offered us, and then deliver those IND enabling studies in the next 12 months. So we are ready to initiate trials as soon as we can in the following year also. So yeah, very rich calendar of events and a lot of value creating milestones, but most importantly, speed in bringing those very important treatments options to patients, which obviously is the paramount objective of Bloomsbury.
Sounds like you've got a very busy 12 months ahead of you. Very exciting. So congrats on the successes so far, and look forward to see what's coming next. And thanks a lot for taking the time today to speak with me about Bloomsbury Genetic Therapies.
Thanks, Laura. Pleasure was all mine.
Thank you. Bye. Bye.